Hello Viking.TV viewers, my name is Damon Stanwell-Smith and today we have a very special issue for you. I am on Viking Octantis on the very last day of our inaugural Antarctic season, and we're going to be talking about the science that we have been undertaking during this amazing time sailing around the Southern Ocean and down to the Antarctic. During the episode, I'll be introducing you to one of the chief scientists of Viking Octantis, Dr Daniel Moore, and also his colleagues on board, who are the scientists that have been undertaking the work with our guests in the field.
Hello, I'm Dr Daniel Moore, a marine biologist with a speciality in population genetics of marine predators. I currently have the privilege of being the chief scientist on board Viking Octantis, on rotation with my colleague, Dr Anna Flink. Welcome to the wet lab on board Viking Octantis. Myself and my colleagues are now going to talk through some exciting projects that we've been running down in Antarctica this season. Working with our partner NOAA, the National Oceanographic and Atmospheric Administration, specifically the US National Weather Service, we will launch regular weather balloons. Down in Antarctica, we launch our weather balloons about every 5 days. However, once we move up to US waters, we'll be launching them every single day. Our weather balloons travel for about 2 hours after launch, and they reach an altitude of about 25 kilometers. As they travel upwards, the radio sound detects the change in altitude, so it starts collecting the data immediately. It collects data on wind speed, wind direction, temperature, humidity and air pressure, and that data is transmitted to our ground station on the top deck of our ship in real time before it's bundled into a package and transferred back to the US National Weather Service. So we're conducting underwater species monitoring, and we're principally interested in the fish species down in Antarctica. This comes from a proposal by the IUCN Species Monitoring Specialist Group, and our lead collaborator for this is Professor Jessica Muick at the University of Western Australia. The way that we're going about our species monitoring is deploying BRUVs, or Baited Remote Underwater Video Systems. Essentially what that is 2 GoPros mounted onto a tripod mount. We deploy these down to the seabed at the end of some long lines and some buoys at the top so we know where they are. We deploy them ideally down to 20 or 30 meters, though if it's a little shallower or a little deeper, that's fine. And we leave them there for an hour. And then we tend to drive the Zodiacs away from where we deployed them afterwards, because we're also interested in the sounds that we record. So we want to just record the reef sounds. Then we'll return and pick them up, and then we review the footage later and back it up to hard drives, which are sent to our collaborators for analysis. The bait that we use to attract fish species in front of our cameras is a species called Atlantic chub mackerel, Scomba coleus. And we have to use that species in particular because it's already in use as a bait fish for the longline fisheries in the Southern Ocean. So we're not introducing anything new to Antarctica.
Phytoplankton make up the base of the marine food web. They are responsible for providing energy to all of the other animals that live in Antarctica and come down to visit Antarctica. And it's important to understand how the base of this food web is changing throughout the entire season from November until March, and to understand how this might change with melting glaciers that come into the coastal environment. Once we're out in the water, we sample plankton two different methods. One is using a phytoplankton net, and the other is just taking a sample of seawater. And the seawater sample can give us a quantitative measure of how many phytoplankton are in that volume of water, as well as the species identification. And then the net is how we concentrate enough biomass that we can preserve that sample to later do DNA sequencing back in the lab. So to get more information about the chemistry and physical properties of the water, we use an instrument called a CTD, or conductivity, temperature, and depth device. And this will profile from the surface as we drop it down to a depth of 60 meters. We're interested in the surface layer where these phytoplankton are living, and we can tell how the temperature and the salinity changes as we go deeper and deeper in the water column. The samples that are collected and preserved for genetic analysis are processed in two ways. So one of the ways is the DNA is extracted. We can get this genetic information to understand which species are present with next generation sequencing techniques. And we can also extract something called RNA, which is sort of understanding the behavior of phytoplankton. So which genes are turned on or off in relation to stress, or regulating osmotic pressure from a changing salinity environment, or different temperatures that they might have experienced. And in this way we can kind of get a measure of what these phytoplankton are experiencing in their environment before we take them out of the water.
So we have samples for microscopy analysis and also for genetic analysis. The microscopic analysis is this quantitative counting of the phytoplankton organisms we saw in the samples. So what we do is to settle a certain amount of sample and count every single organism we found in that sample. So we have a counting of how much organisms of certain species are in that particular place at that particular time. So we expected to see some organisms during the summer, and the first years that we have analyzed so far, we didn't found that kind of organism. Instead, we found some organisms that were really important for the community, were huge quantity of these organisms, kind of million cells per liter, but we couldn't identify them so far, because they are new organisms for science.
Satellites are monitoring changes in Earth and ocean color, and they do that by looking at sea surface salinity, temperature, and chlorophyll, which tells us something about how much phytoplankton there is in the ocean. While we are on the ground, we can also take water samples to get more specific, detailed information, and that can help us ground truth what these satellite sensors are also measuring from space.
The Ferry Box is a piece of oceanographic sampling equipment. It runs automatically in the background without us having to do much of anything at all, and it's really special because it samples several oceanographic parameters. Every 60 seconds we get data input. We get things like temperature, salinity, oxygen, refined oil, turbidity. We even get pigments such as chlorophyll and dissolved organic carbon. We sample seawater because seawater helps drive local weather and global climate by moving temperature and humidity through the atmosphere and around the world. Not only that, but ocean water or seawater responds very quickly to changes in our environment, to changes in climate. And so it gives a feedback loop where it both is affected by and affects climate. In addition to the automatic monitoring that we do with the Ferry Box, we also sample for microplastics. The microplastic sampling is integrated into the Ferry Box. Whenever we want to take a sample, we simply open the valves, let the water flow through 3 metallic filters for a certain amount of time. We usually aim for about 10,000 liters of water. This takes about 10 hours. We turn off the valves and we collect the filters. We concentrate them onto smaller filters, and then we bring them to the lab on board to count the microplastics. We first analyze the samples by simple observation. We want to know what color they are, what large things can we see, and from this we can already get a lot of information. Secondly, we look at them under an optical microscope so that we can get a better idea of what we're seeing. We can zoom in on particles which we'd like to investigate further and get a better idea of their shape, their color, and where they might come from. Interestingly, I wasn't expecting to find many microplastics, as in the research it's thought that the Southern Ocean is very clean in terms of microplastics. Our first samples actually showed quite a few fibers, particularly red and blue, and I was worried that maybe I had contaminated them myself, even though we've tried our best to reduce all contamination possible on board. However, with our collaboration of our resident phytoplankton specialists, they've also seen these fibers, even though they've been filtering water far away from the ship. And so our observations are confirmed and they are real, and we're looking forward to investigating this further.
So citizen science has become an increasingly popular way for scientists to collect data all over the world. This is really important, especially when we look at our remote areas, so our areas like Antarctica that are often difficult to get to and expensive to work in. So utilizing the tourism industry is really important for scientists to collect data they otherwise wouldn't be able to. The really nice thing about citizen science is that anybody can get involved. It doesn't matter how old you are, your level of education, as long as you have a passion for what you do, you can contribute to many scientific projects from all over the world. So Happy Whale is one of the citizen science projects we've been running on board. It's an amazing marine mammal photo ID program. Our main contributors have been of humpback whales, and we've been submitting humpback whale flukes, so their tails, into the Happy Whale platform, and we've been able to assess where these whales have been moving. We had an incredible connection of a whale that was last sighted in 2013, and we re-sighted it in February this year. Happy Whale has over 75,000 individuals registered in their system and an online contribution of 460,000 images. So it's an amazing online platform for us to utilize. Along with this, we have also been testing our Whale ID app. This is in its beta phase. This gives us a real-time association with the whales that we are seeing and what has been recorded in Happy Whale. So it's a really nice, quick way for us to assess if our whales have been registered or not. eBird is another amazing citizen science project that we have on board. It has over 100 million observations contributed annually into a big online platform from birders all over the world. On board, we offer surveys that guests can get involved with, and these surveys contribute towards the distribution and abundance and habitat use of birds seen around the world. So part of eBird, we also have our Merlin Bird ID app. This was developed by our partners, the Cornell Lab of Ornithology, and it's a great tool for our guests to use when they come on board. Essentially, what you do is you take a photograph of any bird, upload it into this app, and it tells you what bird species it is.
What are your reflections on how it's been?
Well, yeah, I joined the ship in January, and it's now getting up to late March. So it has been over 2 months. It's been a long 2 months, in that it's been really tiring. We've been so busy in deploying some of our science operations. And it's been really hard on the day-to-day operations to kind of reflect on where we are in the bigger picture of the science. And there's been a few occasions, particularly in the past voyage or two, where we've done some big summaries, big recaps of the science that we've done. And in putting those recaps together, it's sort of reflecting and going, wow, we've achieved a lot. We've collected a lot of data. We've done an awful lot of different projects, and it's gone really well. And it's really nice to get to the stage where we can look back on that.
Oh, fantastic. And as a visiting scientist, joining for the last 3 weeks, how's it been for you?
For me, I think it's been great because it's also coming down at the end of the season. So looking at that transition between summer and fall, and then really seeing I specialize in studying the phytoplankton community. So being able to see how the end of the season has turned out with the phytoplankton and also getting guests involved in that experience has been awesome.
And you say guest experience, how have they got involved?
In multiple ways, actually, and we've kind of tried to work out where they can get involved at different stages, because what we wanted right from the outset was to make sure that wherever guests are sort of coming into the lab especially, that they are genuinely helping the science. We were looking to avoid that kind of show and tell and make sure that they were really helping us. And so one great example is the BRUVs, our beta remote underwater video systems. At the start of the season, I was preparing all of the bait for those, and it would take me 3 or 4 hours in the lab, sort of smashing up the fish and creating our fish popsicles. And then we realized that what we could do is have guests helping us with that. And so we developed it into a session whereby guests came in, we used the fish as an opportunity to learn about fish anatomy and fish biology, and then we created the fish popsicles together, and it reduced my workload and then therefore helped the science. So that's a great example. And then with deploying the BRUVs, it became very apparent at the start of the season that we couldn't have guests on board the Zodiac we were deploying from. There was just not enough room, and there was perhaps a safety issue. But what we figured out we could do is bring guests alongside in their Zodiacs during their Zodiac cruises, during their landings, and we can hook them up onto the side of our Zodiac, and therefore we can show them as we are deploying and as we are recovering, they can see it in action and see the real way that it happens, and we can talk them through it. So they get to see the preparation in the lab, they get to see actually deploying in the field, and then we've tried within 24 hours wherever possible is review that footage and show it in the hour, so they get to see the outcome of that process.
So the feedback's there.
Yeah, absolutely.
Fantastic.
Yeah.
And with Fjord Photo, your guests have been in the boats with you. How's that been working?
Yeah, Fjord Phyto, the phytoplankton science boat experience, was specifically designed to get people involved putting instruments overboard and getting their hands wet, collecting the phytoplankton themselves. I think everybody who participates so far has said they really love it. It gives them this kind of tangible experience while they're in an environment that's so big and hard to comprehend the scale of the size of Antarctica. And I also think with that element of being able to participate in the actual sample collection helps demystify the process of science. Like what is science? When does it start? When are you actually participating in it? And then it also kind of gives them an opportunity to participate in the legacy of polar science, which is what Antarctica has been preserved for, science and peace. So,
Yeah, I think that's so true, because traditionally science happens in universities and it's kind of in this ivory tower, and on board we're, the lab is surrounded by glass. People can come and just see the science happening, even the behind the scenes stuff all the time, and they're getting involved, and it's kind of breaking down those barriers, and we can all start to create this sense of ownership of the science, which is really, really nice, I think.
And what's the toughest question that you've been asked so far? Particular ones that jump out?
None that I can jump out, but there has been many occasions where there have been former professors at university come and sort of challenge me from their perspective of either, why do we do it in a certain way, or offering different ways that we could analyze the data. And that's been truly fascinating. It's been like almost being at a permanent conference, because our ideas and our methodologies are being challenged in a good, positive way, and forcing us to think about how we do our science differently. And I think it makes it better. So that's really nice to have the Viking audience, the Viking guests on board doing that.
Oh, fantastic.
Yeah.
Have you had any plankton challenges or guests that have asked you difficult questions?
Yeah, I think to that point, there's some people who are maybe more of an expertise in certain areas, and one of the things with the phytoplankton boat was that it was also designed to be the simplest, user-friendly instrumentation that we could find to be able to get good data to answer the questions we have. And sometimes I've had guests wish for more technical things, and I always say, maybe what you did today in the science boat seems very simple, but imagine this same thing is happening over and over and over for 5 months. Then you really build this big picture. So yeah, in that case, it's people wanting more in-depth information. So
absolutely. And big data are so often made up of simple data points being sort of built up. Now we know from our partner NOAA that the National Weather Service say that the weather balloons launched are the most popular in North America event that NOAA runs. That's at least what we were told. How has the weather balloon launch been in terms of popularity?
Our experience has been exactly that. Some voyages we've had almost half of the guests out on deck watching us launch the weather balloons. And I can totally understand that, because it's intrinsically fascinating watching this device sort of disappear into the atmosphere, an area we can't easily go, and getting that data feed sort of in real time that we can then again show to our guests later on. It's been incredibly popular.
That's fantastic. And as you say, one data point builds to the mass to understand something as fundamental as the weather when we're on a ship, climate, which informs so much of the challenges of the modern world and our understanding of how the environment is changing. So we're sitting here having just left Antarctica. We've just seen it sail away. Our guests have just been on the last voyage of the Antarctic season. How is the uniqueness of working in Antarctica, and what's your reflections of Antarctic science?
For me, specializing in polar research, it's apparent that Antarctica is just, in general, compared to the rest of the world, hard to get data from. It's constantly covered in clouds, it's a harsh environment. I love sharing with guests, if they've experienced a really sunny, nice day, that once they experience the winds that come up and the icebergs that can come rip out instrumentation, that it's really a challenging environment in itself to work in. And so I think having the ability to get more data from this region while also engaging people who come to visit is something very special. So as far as I know, there may be in any one given summer 5,000 researchers from all the nations working in Antarctica, whereas you have this visiting community that's coming to the peninsula. And so it's almost like a collective knowledge of the changing system from the beginning of the season when winter ends all the way to when winter begins again. And so to have that engagement has been incredible for the science side and also for the experience.
Yeah. And of course, Antarctica is remote by its nature. The other side of the coin of remote is there's little opportunity to have external support when things are causing challenges. I remember on a previous Viking.TV interview with you discussing how evidence-based decision-making, but there isn't much evidence that evidence-based decision-making actually occurs in the environment very often. And so, as you say, every data point counts.
Yep.
Now, you mentioned the uniqueness of Antarctica and how few observations are made. Have there been any high points or curious things that have occurred in the last 2 months that have particularly caught your interest?
There's been a few. The principal one for me is the observations of giant phantom jellyfish from our submersibles. The species, Stygia gigantea, is principally found in the deep oceans throughout most of the world's oceans apart from the Arctic. And it typically lives between 1,000 and 3,000 meters. And it was first described as a species in 1910. But since 1910, there's only been about 118 recorded sightings ever. And we've seen it 3 times here during the course of the season. So that's a major contribution to our knowledge of the species, particularly as we've seen it at quite shallow depths. And so we're learning something really new about this species, and that's been really fascinating. And it's also an incredible species to see. It's about 10 meters across. And then another thing has been understanding the fish distribution from our BRUBS deployments. There's been some correlations between the type of habitat that they're found in, particularly areas that have lots of macroalgae. So maybe that's useful as a nursery, maybe it's useful for them just to hide from predators. We don't know yet. But also quite a close association between the fish presence and large colonies of penguins too. Again, we need more data to be able to draw that relationship conclusively, but we're starting to get hints just from this first season of, how the habitat underneath Antarctica matters to these distributions.
And I recall in the phytoplankton sampling that you're doing, that you're picking up regular fibres. and that seems to be correlating with other data that's been gathered on the ship. What are you finding there?
So that has been interesting. When one of the samples that we take, we tow a phytoplankton net behind the Zodiac. It's a 20 micron mesh size, and in it, oftentimes when we filter the water out, we've been seeing little fibers that are blue and red. And for the longest time, you know, you think it's my gloves, it's my clothes, it's something, maybe there's static electricity on the net, and so you're rinsing everything as well as you can, and you still see it. And so coming on the ship and having the conversation with Daniel and Brandy and realizing that they're seeing it as well kind of puts this bigger picture in, like, you're not crazy, it's actually something that people are observing. But when you're in an isolated like scientific bubble, you don't necessarily realize, because you're trying to think it might be an artifact, it might be something you're doing wrong in the protocol. But then when you realize it just exists in the water coming from elsewhere.
Really curious, isn't it?
Yeah.
Seeing it coming from the microplastic sampling, and how does that differ from the phytoplankton net tows?
Well, it seems to be quite consistent, you know, across both sampling methods, that we're finding these red and blue fibers. Dr. Brandy Revels has been running the microplastic sampling, and you know, she's been sort of almost tearing her hair out trying to understand, is she contaminating the samples in some way? And so really rigorously trying to make sure that all possible instances of contamination are removed, and she's still finding these samples on the filters. And then to hear that you are finding them too in your phytoplankton nets kind of thinks, oh wow, maybe it's not contamination, maybe they are just out there. And we know from other studies around the world that fibers are really quite prevalent, you know, mainly from washing clothes. They're not filtered out by washing machines currently, or taken out by water treatment plants. So these fibers make their way into our watercourses and out to sea. And it appears, at least from sort of first examination, that they're also really prevalent around Antarctica too.
One of the cool things that can come from this is new questions and new hypotheses for other investigators who might have not realized this is an issue. And so from this, you're also generating more science possibilities. So
absolutely, so watch this space.
Yeah.
So it's fantastic to be talking about what has happened. It's always exciting to think what could happen next. What would your wish list be of things that we can do in the near future with the ship?
I think a real big one for me would be genetics. There is so much potential for eDNA sampling, direct sampling as well. We already have on board a medical PCR laboratory, and so we've proven that that idea of genetics at sea can work, and the science applications of that are potentially huge. A huge number of questions we can answer, a huge number of questions we can formulate from the data we could gather from that.
And I recognise I'm sitting at a table with a pair of geneticists. Do you have any thoughts also on the genetics capacity and where you think that could go?
Yeah, that extends to the phytoplankton as well. For instance, if you're at sea and you can sample a phytoplankton bloom as it's happening and be able to sequence which species are there, then you might even be able to capture other additional samples in real time that you might not have considered if you hadn't have known that it was a rare species that you were seeing or that was detected. And so I think the possibilities of being able to do genetics at sea is incredibly exciting for real-time information.
Absolutely. One of our partners, IUCN, the Species Monitoring Specialist Group, who proposed the BRUVS video recording, have also been very interested in passive acoustic monitoring. Have you had any chances this season yet to deploy hydrophones, and where might that go?
We have. We've deployed them from a Zodiac down to various depths and taken some data recordings, but it's been challenging because we are often recording the sort of Zodiac noise from both our own vessel or from other Zodiacs doing the landings or the Zodiac cruises. Some of that can be filtered out in post-processing of the data, but we need to find another mechanism, I think, of deploying the passive acoustic monitoring so that we can sort of not have the Zodiac noise in the background and get a truer picture of the sounds underneath our ocean.
And I guess our guests are experiencing that not all science works.
That is absolutely true for science. We are creating new knowledge, and when you're pushing at that boundary of knowledge, of course it's going to be difficult, it's going to be easy, not going to be easy, you're going to come across these kind of barriers, and so it's trying to figure out new ways to sort of gain that information, gain that data. It's always going to be challenging, and not everything works first time. That is fundamental to science.
So we've been having great feedback from guests, which is very exciting. How has the science feedback been?
Overwhelmingly positive. The big sort of message that we're getting from our guests is that they want more science. They want to be more involved, helping possibly more. So what we're looking to do at the minute is kind of review our processes, review our projects, and see, are there any more sort of laboratory sessions, field sessions that we can put on where guests can genuinely help us and make our science more efficient and allow us to collect more data. So we're reviewing that and hoping to improve that as we go forward.
Fantastic. And I understand within the Field Photo project that you've actually been asking guests feedback while you've been on the science boat.
We have. So similarly, there's been a lot of positive feedback about how the experience went for people in the science boat. Our project this year has funding from NASA, and one of the elements of receiving federal grants is that you evaluate the impact of your project, and how do you know that people are enjoying the experiencing that the science is being conveyed. And so we actually ask directly for feedback that can help us improve the project in the different elements, but also we ask, have they felt like this has enhanced their voyage down to Antarctica? And overwhelmingly positive, similar response of it has enhanced their learning, what they've learned about the connectedness of the ecosystem, but also what they've learned about phytoplankton and how important phytoplankton are in their role to connect all the animals that exist down here, down in Antarctica.
Well, I hope you've enjoyed having a taster of what we have been doing down in Antarctica. I hope you'll agree also that it has been an extraordinary ride. We're very proud of what we've been able to achieve, and we very much look forward to seeing what this wonderful ship can be doing in the future. We hope to see you sometime sailing somewhere in the remote parts of the world, maybe down in Antarctica or elsewhere. And for now, thank you very much. Until next time.